I'm Kiran and this is Arunesh. We hope to be educating about Flavr Savr tomatoes and give you an idea of how it came to be, how it was used commercially, and what led to its downfall.
Firstly, let's go over some key terms. Genetic splicing refers to the process where the original gene is taken out of the cell and a new DNA is deployed in its place. The process is entirely done with enzymes known as chemical scissors. Depending on the type of restriction enzyme used, different parts of the genetic code can be targeted. A specific restriction enzyme will split apart a specific strand of DNA, leaving a gap in the genetic code. New DNA can then be added in this gap. When a new strand of DNA is added, it takes the binds of the ends of the DNA that was originally cut. They are then repaired. Once the new DNA is in its place, the function of the gene ultimately changes. We can see this through the graphic, that it is a very intricate process and is done individually, cell by cell.
PG, or polygalacturonase, is a gene found in regular tomatoes that contributes to cell wall breakdown. This cell wall breakdown causes cells in general to lose structure and become mushy. This is part of the ripening process and in general is an undesirable quality. When the cell walls are intact, the tomato keeps its shape and doesn't bruise easily, thus meaning the fruit makes it to market intact. This was a gene that needed to be negated during the ripening process to hypothetically create ripe but hard tomatoes.
PG antisense gene. It is a gene that was introduced into tomatoes to halt the degradation of cell walls by stopping the PG gene from taking effect. This antisense gene reduced PG levels in tomatoes to 1% of what they originally were in regular tomatoes. This resulted in firm tomatoes at the market that also were red. This gene was spliced into the DNA of the plant and thus resulted in Flavr Savr tomatoes.
The Flavr Savr tomato was licensed by a company named Calgene, and the gene was isolated and cloned to create the modified fruit. The demand of the modified tomatoes were high due to the increased lifespan and enhanced taste, but still was not enough to be profitable due to high production and distribution costs. The process was designed to keep the fruit harder and more attractive to sell at the market. Another company called Zeneca introduced tomato paste from these GM tomatoes, selling more than 1.8 million cans in the UK alone. However, due to some customers' misplaced accusations of compromised food safety, these products had to be taken off the shelves by 1997, despite food safety not being harmed or proven to be harmed. Dr. Arpad Putsai conducted from his experiment on rats by feeding them GM potatoes that they had biological effects that may have come from this model.
A gene was identified by the original company Calgene, which was found to stop the ripening process. This gene is called PG and has been found to contribute to the breakdown of cell walls. To stop this process from taking place and thus keeping the fruit harder, an antisense gene was created in order to stop the breakdown of cell walls in the fruit. As we can see from the diagram, the natural sense, or PG, would eventually lead to proteins that would result in the decay of the quality of the tomato. In contrast, we can see from the bottom half of the diagram, when the antisense gene is genetically spliced into the cell, it would code for complementary antisense mRNA and would result in the tomato to maintain its desirable shape and color. The two strands, as seen in the diagram, are stuck together, not allowing the PG gene to be produced, exponentially slowing the ripening process. This antisense gene was so successful that levels of the PG gene in these GM tomatoes dropped to under 1% of levels found in traditional tomatoes. This relates to our genetics unit, as we've covered genetic modification and the different methods conducted, as well as the physical and ethical issues surrounding the tomatoes.
From this chart, we can see the true power of the antisense gene and its sole importance in preserving the life of the tomato. It is evident that in an ordinary tomato, which has had no tampering with chemicals or genetic modification, it has an average market life of 10 days before it starts to degrade quite badly. Within 20 days, the tomato has mostly lost its shape, taste, and its ripeness. It is much softer than a tomato that has been in the market for 10 days and would be considered by most supermarkets to be waste and would be thrown out. By 45 days, the tomato would have become highly rotten and unpleasant and would not be consumed by any humans. On the contrary, Flavr Savr tomatoes have a seemingly longer lifespan, and due to its lack of expression of the PG gene, it will be ripe and maintain its shape for a great period of time. It is clear from the chart that after 20 and 45 days, the tomato is very similar in terms of colour, freshness and shape.
Discovery was significant and useful in a number of ways. It was the first fruit to be tested and successfully genetically modified in a small scale. This was promising as it showed that tomatoes, as well as other fruits and vegetables, can be modified to suit the people's need. It was a pioneer in the commercial market for GM crops and had inspired scientists and biotechnologists to test different modification of crops like corn and soy to be able to be resistant to pesticides or pests themselves. It was the first real test and the first company of its kind in isolating a gene, splicing it into the correct DNA sequence to grow a crop from it. It showed there was untapped potential in the market. For business, it would save the need for them to continually buy new stock. This would lead into supermarkets increasing profits. For consumers, these tomatoes could be stored longer, so there was no need to use them immediately, and were also enhanced in taste and flavour.
All demographics of age and sex would be affected by the discovery of Flavr Savr tomatoes. In Australia, over 8 million kilograms of tomatoes were sold in every 3-month period and has been considered a staple in worldwide cuisine. In between the era of Flavr Savr tomatoes between 1994 and 1997, there was 5.4 million kilograms of Flavr Savr tomatoes sold and were approximately 12% of all the tomatoes produced worldwide. The biggest demographic of buyers were a bunch of elite restaurants who were keen on enhancing their food, purchasing 2 to 2.6 million kilograms over its existence. The next biggest buyer were a small bunch of manufacturing companies who were interested in increasing the longevity of their products, such as Heinz, who were reported to buy 1.5 to 1.7 million kilograms. This proved that even if people could not afford to consistently buy GM products, they were consuming it through packaged food or restaurants. Everyday customers bought around 1.8 million kilograms of these GM tomatoes over its time. Another interesting demographic that were affected were genetic scientists. The services of effectively duplicating was in high demand and would mean more of these professionals needed to be employed. From the mid-1980s, there was 80% more people to employ to meet the demand of customers. There were a number of rich businesses who wanted to invest in the crops and would lead to 200 more investors on the stock market for research companies responsible.
Three major issues that surrounded this discovery is the question of the ecosystem being harmed, the potential of there being a superbug, and could there be cross-pollination with other plants. The ecosystem theoretically can be harmed, as there could be genes inserted into the fruit that harm pests and thus killing them and harming the food web and ecosystem. However, in the case of Flavr Savr tomatoes, it only inserts a gene to stop PG production, so the ecosystem shouldn't be affected. Another issue in the gene is the gene creating antibiotic resistance in bacteria and thus creating a superbug. This could be a threat, as there is a gene that promotes cell walls staying intact, opposite to the function of antibiotics. However, due to the gene being designed for plant cell walls, this shouldn't be a great issue. The third issue raised is the potential of cross-pollination. This would be an issue, but the company controlled the growing zones to make sure that the Flavr Savr tomatoes were the only plants grown in their region. However, if it were to get out, this could pose a problem. This is an article by California Agriculture talking about the Flavr Savr tomato, how it came to be and what happened to it. The article was published in July 1st, 2000 by G Brewing and JM Lyons. The nature of this article is a website article/report.
In conclusion, GM tomatoes are a pioneering breakthrough in the field of GM crops and have led to the discovery of other GM crops such as corn and soy, which are currently almost 90% GM modified. These crops are now being consumed safely by people all around the world, and as a result of Calgene's breakthrough, we have other GM crops being developed today. For better or worse, it is still a topic for debate, but there is great potential in this field.
Thank you for watching, Mr. Cornell.
Okay. Mr. Cornell, set eye contact. St Kilda Saints are a crap football team with crap players and a crap coach.